Okay, let's take it from the top one more time, just so you will get this down. The notion to make a motion is a voluntary thing. It is executed according to the conscious will of the person. So that begins with the pyramidal cells of the motor cortex. Those action potentials descend from the cerebral cortex down through the internal capsule. They are then seen crossing over at the decussation of the pyramids and the medulla oblongata. From there, they descend down the spinal cord in what are called the corticospinal tracts. So that is one neuron. That is called the upper motor neuron. It will synapse with a lower motor neuron as it exits the spinal cord and the fiber enters into one of the peripheral nerves. So we have an action potential, which is a wave of depolarization of nerve cells, two nerve cells in the motor pathway, upper motor neuron, lower motor neuron, reaching the neuromuscular junction. The action potential or wave of depolarization causes calcium channels to open at the synaptic knob. Calcium rushes in, and as that happens, the neurotransmitter is released into the synaptic cleft. The neurotransmitter that excites skeletal muscle is acetylcholine. So skeletal muscle tissue has receptors for acetylcholine. When the acetylcholine binds to the receptor, sodium channels are opened in the sarcolemma. When the sodium enters into the muscle tissue itself, it depolarizes the muscle cell membrane, which we call a sarcolemma. When the wave of depolarization drifts down into the T tubules, those T tubules come near to the sarcoplasmic reticulum, which is a storage location for calcium. When that wave of depolarization passes by the sarcoplasmic reticulum, it releases calcium into the sarcoplasm. That free calcium in the sarcoplasm will then bind to a regulatory protein called troponin. When the calcium binds to the troponin, it causes the troponin tropomyosin complex to move. When that complex is moved, the binding site on the actin filament is exposed. When the binding site on the actin filament is exposed, the myosin heads can attach to that binding site. Now, where do we get energy? The myosin head cannot pull the actin strand. I think the example I gave was a person using both hands to pull a rope. So the rope is attached to a boat somewhere and you want to pull the boat to shore. Well, you pull with one hand, then you reach with the other and pull with it. And this is how the myosin heads pull on the actin filaments, which draws Z disc closer to Z disc. So at the microfilament level, that is contraction. So all of those sarcomeres combined performing that particular action results in contraction of the muscle cell. Now, we haven't talked about how we get this done because this is an energy requiring dynamic. In order to cock the myosin head, we have to spend a molecule of ATP. Also, to put the calcium back into the sarcoplasmic reticulum requires the expenditure of ATP. So we're not going to get contraction of muscle unless we're willing to spend some of our energy. And the energy that we use to power cellular activities is adenosine triphosphate. Okay, so that is bringing you to where we had gotten in the previous PowerPoint, and I will pick up there and move along. So let's go to slide 2. So what is inside of a skeletal muscle cell? Well, it is chock full of those sarcomeres, which are actin and myosin and elastin and titin filaments. The actin and myosin being the contractile proteins, the troponin and tropomyosin being regulatory proteins. So skeletal muscle cells are chock full of those little contractile units. But for those contractile units to do anything, there has to be an energy source. So also in the muscle cell, we have oxygen in storage. You may be familiar with hemoglobin. Hemoglobin binds oxygen and transports it in the blood. Turns out that muscle cells have hemoglobin in them. Since there is hemoglobin in there, then we have the possibility of binding oxygen inside the muscle cell. We don't call it hemoglobin, we call it myoglobin. Myo just means muscle. So what it is a way of binding and storing oxygen inside the muscle cell. So muscle cells have myoglobin, that is oxygen in storage. Well, the reason we want that is because we use oxygen to burn organic molecules such as glucose or fatty acids. So oxygen is the fire, glucose is the wood. Well, there has to be an organelle where that takes place. That organelle is called a mitochondrion. So the mitochondrion is the fireplace. So we have oxygen available, courtesy of myoglobin. We have glucose inside of muscle cells. It is a storage form of glucose that is called glycogen. And we have the mitochondria, which has the unique ability to take the oxygen and burn the glucose and therefore release energy, which can be used to make ATP from ADP. So this is one source of ATP for the muscle cell. But there is more to it than that, because when a person begins to exercise, there is only a certain amount of myoglobin inside the cell. There is only a certain amount of glycogen inside the cell. So looking at this diagram on this page, which is a little too abbreviated for my blood, but all the same, I think you can get the point. If a person initiates an intense exercise activity, such as taking off running full speed, as if there is an armadillo with rabies chasing after you behind, well, you take off running as fast as you can go. You're going to use up the available ATP in the muscle cells themselves within just a few seconds. So the number given here is the ATP stored in muscle cells is used up in 6 seconds. That's about right. Okay, what are we going to do? The armadillo is still coming after us, and he's foaming at the mouth with rabies, and he's snapping at your behind as you run full speed. Well, you done exhausted all of your available ATP. Well, we can make ATP by what is called the phosphagen system. We can borrow a phosphate group from ADP, that is adenosine diphosphate. Well, it has 2 phosphate groups. So what we do is borrow one. So we borrow the phosphate group from ADP to make a molecule of ATP. So we can keep going No stopping here. We've only been running for 6 seconds. Another source of phosphate groups is a molecule called creatine phosphate. So creatine phosphate is in the muscle cell, and when we need a phosphate group to add to our ADP and therefore make ATP, we will borrow a phosphate group from creatine phosphate. Mission accomplished. We were able to make some ATP and therefore keep going. So that is called the phosphagen system, and the phosphagen system might be good for another 10 seconds of intense exercise. Well, we're going to have to go beyond that. We've only been going for 16 seconds, and the armadillo is running faster and faster, trying to chase your behind down. And so you got to keep going. You got to have a source of energy. Well, we do have that glycogen in storage, so we will break the glycogen down to glucose molecules, and we don't have any oxygen to burn it. We used up all of our oxygen in the first 5 or 6 seconds. So here we have glycogen that is a source of energy, but we don't have a way to burn it because we don't have any oxygen available. The myoglobin supply of oxygen expired, so we have to ferment the glucose. Fermentation doesn't require oxygen. When we ferment glucose, no oxygen is required. Some ATP is formed, not much. We don't form a whole lot of ATP. I think it's 2 molecules of ATP per molecule of glucose when we ferment the glucose, which is what we're going to have to do here because we don't have oxygen available. So we ferment. The other problem that goes with that is when you ferment glucose, you end up forming lactic acid. Did I say lactic acid? Well, that's acidic, and it's going to contribute to fatigue. So an accumulation of lactic acid is what makes the muscles begin to burn, and they begin to wear out. So here in the 30 to 40-second time period, what we're doing is fermenting the available glucose. Well, the lactic acid is going to accumulate when you do that, and fatigue begins to catch up with you at about that point. Fortunately, the heart and lungs catch on to the need for more oxygen and more glucose to be delivered to the muscles. So we dilate the capillaries going to the muscles. The heart beats faster, it beats harder. The lungs take in more oxygen and they eliminate more carbon dioxide. So at about the 40-second time of intense exercise, the heart and lungs begin to catch up to the demand, supplying the blood, which has the oxygen, which also has the glucose, to the skeletal muscle so we can stop fermenting and start oxidizing glucose. Oxidation of glucose gives us 38 molecules of ATP per molecule of glucose. There is no lactic acid accumulation when we oxidize our glucose. So we're able to keep running beyond the 40 or 50-second time because the heart and lungs will catch up. Now, how long can you go beyond that? Well, it depends upon your cardiovascular health and how how efficiently your lungs can remove carbon dioxide from the blood. So that comes with training. So you get better at it the more you challenge yourself. So this is the energy to run from the armadillo with rabies, because certainly in this world, there's got to be one chasing you down from somewhere. Next slide, please. Well, if you take off contracting any skeletal muscle very quickly and against any resistance, then you're very familiar with the thing called fatigue. So you can take a dumbbell. It may not weigh 15 pounds, but if you do curls with that dumbbell, about the time you get to number 20, you're going to feel burning in your biceps. That is the fatigue setting in. And why does that happen? Well, that happens because of the accumulation of lactic acid, which lowers the pH of the environment. Proteins are pH sensitive. We're depending upon the proteins, actin and myosin, troponin and tropomyosin. These are the proteins involved in muscle contraction. We're depending on them to do what they can do in order to contract our muscles, while at the same time, we are decreasing the pH of our body fluids by the accumulation of lactic acid. Proteins are pH sensitive. They have a pH at which they work well. If you change that pH, they're not going to work as well as they did before. So we are creating an acidic environment, and we're expecting our actin and myosin to do what only they can do. They don't work very well when there's an accumulation of lactic acid and therefore a drop in pH. So that is one of the explanations for fatigue. But there's more to it than that. What is listed on this illustration is just the accumulation of lactic acid. There's more to fatigue than just that. There is a thing called junctional fatigue. If you continue running beyond, say, 20 miles, which I know some of you do this, you run and you run and you run. Some of you run more in one day than I run in a year. When you continue to push your limits, you can come to a thing called junctional fatigue. It isn't an accumulation of lactic acid. Your heart and lungs are healthy enough to supply the oxygen and the glucose that you need to oxidize glucose. So we don't have a lactic acid problem, but all the same, you experience fatigue. Well, that is called junctional fatigue. Remember, we depend upon the acetylcholine to stimulate the muscle. Well, we only have a certain amount of acetylcholine. So we can come to a place after a prolonged period of hard work where we face junctional fatigue, the depletion of acetylcholine necessary to stimulate the skeletal muscles. So the legs begin to get very rubbery, and you might have a hard time staying up. Beyond that, there is a thing called CNS fatigue. Remember, we are dependent upon signals coming from the brain and traveling down the spinal cord to get to the muscle. Well, what if we deplete our ability to transmit that signal? That signal has to go from one neuron to the next by way of neurotransmitters, and then from the second neuron to the muscle by way of the acetylcholine. So after prolonged exercise, we're not talking about a lactic acid problem. We may have made it through the junctional fatigue. The brain grows weary of sending the signal. So if you grow weary of sending the signal to contract a muscle, then the muscle doesn't contract. So that is called CNS fatigue. And only those of you who exercise outrageously, I mean, these people that run in marathons and things like that are the types that I'm talking about. All of the rest of us would never experience junctional fatigue. None of the rest of us would ever experience CNS fatigue. But people who take on these enormous physical challenges can probably testify. Yeah, there's something here besides just the muscles burning. Something begins to fail at about mile 20, and something begins to really fail at about mile 26. So those are explained by junctional fatigue and CNS fatigue, which is different from. lactic acid fatigue. All right, on the other side, we're talking about muscle soreness, and only one explanation for that is given here. When muscles have not worked for a while and you go to the gym or you go out on the street or wherever it is, you're going to do some intense work. The muscle is not prepared for the challenge that you place on it, and it can cause the actin and myosin heads to be overly stressed. That is one explanation for muscle soreness. Also, remember the actin filaments, by way of a protein called dystrophin, attach to the cell membrane. Well, if things are not prepared for it, then the dystrophin can tear the actual cell membrane. And so you have what's listed here as micro tears in the muscle from strenuous exercise. So the only way to avoid muscle soreness is to do what you do regularly. If you do like I do, you take one day off, and then that one day becomes 20 years, and you go out and you try to run 6 miles. Man, I tell you, you're going to have the shin splints like you ain't never heard of. Can I get a witness? Okay, so these are some of the explanations for fatigue and muscle soreness. I'm sure that there's probably more to it than this, but this is a pretty good start. Next slide. Well, as you can tell by looking at a photograph of this Boston butt roast, we actually have two types of muscle, and you can see that one area is reddish, the other area is kind of white. So what we have then is a difference between the type of muscle that a person might have. Now, all people have red and all people have white. So it's a matter of the ratio between the two. So some people have a greater abundance of red muscle fibers. Some people have a greater abundance of white muscle fibers. Well, the difference between the two is this. Red muscle has many, many sarcomeres, and the actin and myosin filaments are very large and thick. So when you have many sarcomeres and the actin and myosin filaments are very big and robust, then that muscle will generate a lot of strength. So that is short-term, high strength, low endurance. The person who has mostly red muscle tissue can run very fast, but they can't run very far. They can jump very high, or they can push an enormous amount of weight, but they can't do it very many times. So red muscle tissue is extremely strong, but it is very low in endurance. On the other side, we have the white. Well, this is muscle tissue where the sarcomeres are not so plentiful and the actin and myosin filaments are not so large, but that muscle tissue is well supplied with capillaries. The muscle cells themselves have a lot of myoglobin, and they have a lot of glycogen, and they have a great number of mitochondria. So in the white muscle tissue type, you've got a lot of blood, you've got a lot of oxygen, courtesy of the myoglobin, you've got a lot of glucose, courtesy of the glycogen, and you've got all the fireplaces you need. Well, the sarcomeres are very small and kind of scrawny, so this person is not very strong. This person is not going to win the Olympic 100-meter dash. This person is not going to be a world competitive bench presser, but this person can keep it going and keep it going and keep it going because they don't fatigue. They have all the mitochondria. They have the blood supply by way of the capillaries. They have the myoglobin, which is oxygen in storage, and they have the glycogen, which is glucose in storage. So what you have is a person who has a high percentage of white muscle. Well, then they are the marathon runners. They can run for miles and miles and miles, or they can endure an athletic event for hours and hours, whereas the red muscle person, they can knock your head off with one punch. But if you make them go 15 rounds, chances are they're going to fall on their face from fatigue. So this is well illustrated in boxers. You have some boxers that are enormously strong. George Foreman was one of those, a very strong boxer. Usually, George's fights didn't last beyond 2 or 3 rounds because he would destroy the opponent with his powerful punches. Well, Muhammad Ali met up with George Foreman down in Kinshasa, Zaire, and Muhammad Ali was equipped to go the distance. So he didn't have the punching power that George Foreman had, but he had the endurance. So what he did was allow George to just punch on him for about the first 4 or 5 rounds, and George became so tired that he couldn't hold his hands up. And after that, Muhammad Ali took it to his face. So it's a difference between the proportion. Some people have more red, they are very strong. Some people have more white, they have tremendous endurance. So that's the two types of muscle based on the availability of the resources needed to make ATP or the resources needed to contract very forcefully. Next slide, please. So here we have a muscle that contracts every zero point 0.8 seconds from the time a person is only about 21 days past conception until the day when 6 guys in black coats show up. That muscle contracts every zero point 0.8 seconds, waking, sleeping, rising up, lying down, texting and driving. That muscle contracts 75 to 80 times per minute without resting. If the heart decided to rest, then you also will rest in peace. I thought that was pretty funny. All right, so how is it that the heart muscle can work with such endurance that it will completely contract every zero point 0.8 seconds for all the years of our lives? Well, the cardiocytes are well equipped with myoglobin. Yes, that's our supply of oxygen. The heart is not allowed to fatigue. Now remember, we fatigue when we ferment glucose. We ferment glucose because oxygen is not available. Well, we can't take that chance here, so we have much myoglobin inside of the heart muscle cells. We have much glycogen. We cannot do without our ATP, and we're not allowed to ferment. If the heart muscle begins to ferment, the lactic acid accumulates in the heart muscle cells and the heart begins to hurt. That's called angina pectoris. The heart muscle is hurting because the lactic acid accumulates in the cells, and acidity is never something that cells need. So we're not allowed to ferment. Your chest is going to hurt if you start fermenting your glucose. So we have much myoglobin, we have much glycogen in storage, and we have enormous and many mitochondria. So the heart muscle cell is well equipped to oxidize organic molecules. Oddly enough, at rest, the heart muscle cell prefers to oxidize fatty acids. But when we exercise, the heart will shift over to oxidizing glucose. But at rest, for whatever reason, I don't know, that anybody knows, the mitochondria prefer to burn fatty acids. So what we have then is heart muscle tissue that is made for the purpose of contracting every zero point 0.8 seconds for whatever number of years a person might live without ever generating lactic acid, without ever fatiguing or without ever doing without the things that it needs. So it's well supplied with glycogen, myoglobin, mitochondria, and obviously there is a rich blood supply since all of the body's blood passes through the heart per minute. That's right. The blood that leaves the heart at 12 noon returns to the heart by 12:01. So the heart has a good blood supply because the blood has to pass right through the heart. Well, that also depends upon the health of the coronary arteries. So the coronary arteries, if they are blocked, and they do become blocked by various things such as fatty plaque or sometimes by a blood clot. If the coronary arteries, which deliver red, oxygen-rich blood to the heart muscle itself, if there is a blockage, then those cells do not get the oxygen they need. If they don't get the oxygen they need, they ferment. If they ferment, they generate lactic acid. If that happens, the chest starts hurting. That's angina pectoris. And what they say in clinical talk is time is muscle. Well, that's right. So from the time that the chest starts hurting until the time that the cells start dying, that can be measured in minutes. So you don't want your heart muscle cells to die due to a lack of oxygen. All right, we'll talk about that when we get to heart physiology another day. Well, notice first of all that the smooth muscle cell is not like a skeletal muscle fiber. Smooth muscle cells are normal in their size. Skeletal muscle fibers are very thin and long. Also, smooth muscle tissue has one centrally located nucleus. Notice that the sarcomeres, which are labeled here as thick filaments and intermediate filaments, that is actin and myosin, they don't run parallel to each other, so they don't overlap. Well, if they don't overlap, then there are no striations, because the striations that we see in skeletal muscle and cardiac muscle are due to the overlapping of the thick and thin filaments. Also, smooth muscle cells have no T tubules. Their sarcoplasmic reticulum is very minimal in its contribution. There's very little calcium that is stored there. So most of the calcium that is going to cause the smooth muscle cell to contract has to come in from the extracellular fluid. Well, that's going to take a little more time than what would happen if the calcium were released from within the cell. Also, smooth muscle cells have no troponin and they have no tropomyosin. So when the calcium does come into the smooth muscle cell, it binds to a different regulatory protein called calmodulin. So there are some differences in the anatomy and some differences in the physiology with smooth muscle. Now, as far as contractile strength goes, we generally think of smooth muscle tissue exerting a slow, sustained, and relatively weak contraction. Well, that is not the case with the uterus. When it's time for the baby to be born, the uterus can deliver a very forceful contraction, which will force the baby into and out of the birth canal. Okay, so these are some of the characteristics of smooth muscle tissue. such that we would have in the walls of the bronchi, in the walls of the arteries, in the walls of the stomach, in the walls of the intestines, in the walls of the bladder. So we have smooth muscle tissue distributed throughout the body. And one other thing, I have mentioned this before, smooth muscle can be stimulated to contract by nervous innervation, that is autonomic nervous system functions. Also, smooth muscle tissue is stimulated to contract by stretching it. That's one of the most common ways that smooth muscle is stimulated to contract. We have hormones. It is the oxytocin that is released by the posterior pituitary gland that stimulates the uterus to contract at the time of labor and delivery. And also there are some chemical irritants, some chemicals that when they come in contact with smooth muscle tissue, the smooth muscle will respond by contracting. So those are called irritants. An example of that is X-Lax for treatment of constipation. The way the X-Lax works is by irritating the smooth muscle tissue of the large intestine, and because of that irritation, the smooth muscle tissue contracts. So mission accomplished. Okay, that's all I have for this, and we will move along to the several slides of muscle anatomy after this.